Hello?
Mhm.
Oh, you can? Okay. Um, well, anyways, welcome. Um, Not sure if anyone wanted to mention anything before we start, or there he is.
Oh,
Hi, how are you? Yeah, there you go.
yeah, I guess it is the browser.
Yeah. Um, yeah, I hear you and Hussein are making progress on the embryo or the breast cancer
Uh,
physics.
yeah, yeah. He seems to be coding, but I haven't seen the results from the code yet.
Oh, okay. Yeah.
Okay,
Yeah.
okay. I sent you that problem with the textures.
Right.
I think there's a general problem there.
Okay.
You know that if you take a Perlin noise in 3D and project it, is the projection Perlin noise? I don't know.
Yeah.
Or if you take a projection and it has Perlin noise, does it come from 3D Perlin noise?
Right.
Okay, and I have a sneaky suspicion that this reversibility is only true for Gaussian noise.
Okay.
Okay, because Gaussians seem to transform between dimensions. Fine. So I leave you with that puzzle.
Yeah, I'll have to look into it. I haven't had time lately, but
Okay, yeah. Well, I think we'll make the deadline for the book. The Russian group is also working now, and they've come up with a clever way of spreading the lines so that you don't miss any tumors of a given size.
yeah,
Okay. Okay. So, they have produced a little bit so far, but not a full algorithm.
right, right. Because you could miss tumors within those spaces when you sample.
Yeah. Right.
Yeah.
Right. And they've come up with, as I said, with a way of generating the lines. So the way they put it, so you hit each tumor at least with one line.
Oh, okay.
Okay. Now, the method that Hussein are working on the other hand, is a greedy algorithm. If you hit a tumor, you go after it, and you sort of ignore the rest.
Yeah.
Okay. Uh, so we'll see which does best. It's not obvious.
Yeah. Sounds
Okay,
good.
but
Yeah.
it's sort of as I figured that there will be alternative algorithms.
Right.
Okay, and the other thing is they seem to be taking an image independent view, whereas Hussein and I are doing very image dependent algorithm.
Yeah.
Okay, so that's the current state of things. Uh, Hussein seems to be getting more and more involved in this, and so I'm sending him some general CT algorithm papers as we go.
Yeah. Yeah. He seems to be pretty excited about it. So.
Yeah, yeah, yeah. You may be rich someday if he gets his startup going. I doubt I'll be alive, so I'm not
Yeah.
worried about it. Anyway, besides, there's nothing to spend it on here. But now
Oh.
I'm getting an echo.
Yeah.
This town is the town I'm in is down to uh, only one business, and that's the post office.
Oh. Yeah.
We had two, but that was a veterinarian's office, but he died in July.
Oh.
So that's finished.
Yeah.
Uh, But so wealth doesn't matter here.
Yeah. Yeah. Yeah.
Mhm.
Well, thanks for the update. That's good.
Okay,
Yeah.
strange update.
So I don't know if Morgan has got anything. I have some things actually from his posting. Um, we'll go over that a little bit. Yeah, so why don't we just actually, I'll go over another thing here. So let me share my screen. I'll go over another thing first. So this week, Paki Bonchalia, who was one of our Google Summer of Code students, she published a blog post on her project, and this is the result. So this is developing adaptive neural networks, growing graphs for dynamic data representation. And I'll post it in the chat. Hello, Jesse. So this is the blog post she prepared for her project. It basically goes over the project. So, again, this was a project for Google Summer of Code. There were two parts to this. There was the topological data analysis using KMapper. This was a way to sort of explore the data for complex data structures, things that you might expect in a network topology. And then the second one was growing graph neural networks, where that was the main part of the project. So actually, I think Himanshu Shogul, I think it was last year, worked on the TDA using KMapper. He kind of played around with it a little bit at the end of his project, but didn't really get very far. And uh, Paki also did this at the beginning of her project, but there's a lot more to do here. I don't really know if we could find, like, I don't know if that could be like the focus of a project, just that or what, but I'm really intrigued by some of the work that's been produced. You just need to kind of go in a more formal direction for that. Thinking about topological data analysis and what we can get out of it. People have done this with C. elegans connectome already, so there is some precedence for it. But otherwise, I'm kind of interested in that aspect of the project. And although she did work on that, it wasn't to any sort of completion. So that's still open. But the more important part is this growing the graph neural network part, which I think is going to be really interesting. So this K-mapper algorithm. So this is basically a way to sort of explore the data. So this is using topological data analysis, using the K-mapper implementation, which is where we can visualize high-dimensional data and identify key topological features. So this is a Mathematica applet that allows you to do K-mapper, and so it looks kind of like maybe like a UMAP, but it's not a UMAP. And so, what it does is basically plots the data out in certain ways. And with topological data analysis, allows you to do is find like shapes in the data. So the shape will have the data will have a certain shape, especially if you reduce the dimensionality, and you can plot it in different ways and see the shape. And you can actually analyze it using more advanced techniques. So this is the visualization step where you see it in the applet here. But there is like a mathematics underneath, and that's something that's, I think, something to follow up on. Now the shape of the data is interesting because it gives you, if you think about like a trend line, a linear trend line, a linear regression, it's like something that follows a line. So it's like a point of clouds that goes along a line. Or, if you think about like in classic topology, you have these, annulus like shapes that look like a donut. And sometimes data look like that. Sometimes it's because it's distributed in space. Sometimes it's because it's distributed in time. Sometimes there are other features that give it that shape. But the idea here is we want to be able to find some shape to the data that isn't like the anatomy. So this is sort of the summary of this. Um, and again, like there's probably more to do here. I think Pocky did work on this for a while during the summer, but again, I'd like to explore this more. But this allowed us to go to sort of the next step, which was growing the graph neural network. So that Mehul did some work on hypergraphs that were graph neural networks, and so Pocky was just doing a standard graph neural network where we had this growing network. So the idea is that in an embryo, you have a lineage tree. The lineage tree unfolds by having a single cell that divides and does this repeatedly until you get to differentiated cells. So that produces this sort of growing graph. If you plot the lineage tree out as a complex graph or a complex network, you get these. The number of nodes increases. Basically, there's a doubling of nodes, and so you have to sort of readjust the network uh, as that happens. But you also have to visualize that, and so that's where this kind of project is useful. So we had a couple things in the growing graph neural network. We had temporal node addition, which is where the graph reads from a data set. So every time a new cell is born, it's added to the graph. Every time a cell dies, it's removed from the graph. And so you end up basically with this doubling of nodes every so often, and then you have this change in connectivity. Um, the second part is nodes are dynamically added to the graph at their specified birth time. So the timing of this process is such that it mirrors like what happens in an embryo. And then this simulates cell division. Um, and oh, you were going to say something?
Uh, yeah, Bradley, one minor correction.
Okay.
Bradley, a minor correction. In the differentiation tree, we refer to each intermediate cell type as if it is differentiated.
Okay.
Okay, so that it's not just there's terminal differentiation.
Okay. Yeah. Yeah. I mean, this was just simplifying it. So yeah, you would have like
Yeah,
these different cell types that kind of are intermediate stages.
yeah. Most people count those intermediate cell types as differentiated.
Okay.
Okay.
All right. Then okay. So that was terminal node addition, where we have this process of adding and removing nodes. Then we have this spatial learning process, which is where we just don't want to just add nodes. We want to learn spatial relationships between nodes. So basically, the daughter cells are in some spatial relationship with each other. And this is going to be important for like things like looking at polarity and looking at other structures. Like we get these intermediate cell types that start to form some sort of structure within the embryo. So in the C. elegans, you have this comma phase of development where the embryo basically forms a comma, and where there's like an indent in the middle, and so we can look at those sorts of things, those spatial relationships. The data not only have the division information, but also some spatial location information as well. So that's good. Uh, the network computes and optimizes the distances between every pair of nodes, and this was done using the CMA-ES algorithm. So this is an evolutionary algorithm. So this was forked from a project where they were doing this with neural networks, where they were growing neural networks. And so they have this evolutionary algorithm that's associated with that source of the fork that kind of it walks through this sort of optimization process. So you can see that you're basically you're using an evolution strategy to initialize the optimizer. You're performing this optimization loop, so it looks for um, has like a loss function, and it looks for the best answer from that, and then it's retrieving the optimized parameters. So it's basically optimizing this whole process of spatial learning using this algorithm. The third part is biological analogy. So this can be likened to embryonic development, although it's not quite the same thing. We're just kind of abstracting it according to some sort of complex network structure. And then the graph structure evolves both temporally through these time node additions and spatially through learning the internode distances and kind of making sure that they're, you know, in a reasonable sort of arrangement. So this is a GIF that shows kind of how one of these grow. This network grows. Kind of it's expanding, and the way it's kind of plotted out, it's weird because every time you plot new cells or every time the network grows, it's visualized in a different way. And this is one of the things about complex networks and working with them is that these layouts are kind of hard to optimize and get exactly the way you want them. So, like if you want to see, for example, you see that you can't see a lot of the labels are all smushed up. Some of the cells are all, or the nodes are all smushed up in blue. That's because it's just visualized in a way that is hard to see. So, you could visualize these in a circle, like a lot of people use, circular layouts, but that doesn't really give you, that spatial information. If we use something like this with spatial information, we end up with things that are bunched up in certain places and you can't really see the fine structure. So, if we were doing this in a live app, we could zoom in on a certain region or rotate the three-dimensional graph and maybe see one perspective on this. But this is this is a problem in complex networks in trying to interpret sort of their structure by kind of visualizing the nodes and their connections. So we have like things like force directed layouts, circular layouts, these kind of layouts where you keep shifting the sort of the orientation and the topology, but it's basically the same degree of connectivity. And that's the best we have to work with. So that's one of the problems with these kind of structures is it's kind of hard to get any insight out of just visualizing them a certain way. So, or maybe the best way you have, the easiest way you can visualize them. So this is, that's maybe why we use or where we're interested in the topological data analysis. So we can understand if there are any structures in there that are not easy to see from something like this. The other thing too, are of course we can now calculate graph statistics. So we can calculate things like centrality or graph distance, the span of the graph across the diameter of the graph, and other types of things. So we have a number of ways we can describe this structure, these growing structures, that we can't do with say just a normal embryo, or some sort of visualization procedure, or segmenting the cells out and analyzing each cell, or even with a lineage tree where we're just kind of looking down sort of a directed branching structure. So that's what we have with that. So again, challenges and learnings. Growing a GNN came with its unique set of challenges. We wanted to design the training loop. So, determining an effective training loop for the network. So, in other words, there was some source data that was embryo data, and that was plugged into this algorithm, and then the optimization procedure happened, and then you get this visualization. So, getting more data, of course, I need to send Taki more data. Um, we also need to work on, some of these aspects of optimizing the training. So, maybe training the data set or training the model with other data sets. Maybe that are not C. elegans. It may help. It may generalize. It may not help. Okay. Um, so then there's the limits of backpropagation. So standard backpropagation, a key component of neural network training, proved inadequate for the growing GNN. So, we usually use backpropagation in a lot of neural network applications, but in this case, we couldn't do that. The increasing number of parameters as the network grew made traditional backpropagation ineffective. The nonlinear growth pattern of the network further complicated the use of gradient-based optimization methods. So this is again the solution is this algorithm CMAES or covariance matrix adaptation evolution strategy. So this is what comes in the thing that was forked. This is although it's a powerful optimization algorithm, it doesn't rely on gradient information. But it's also particularly well suited for nonlinear, non-convex optimization. So this is a non-convex problem, nonlinear problem, and we can use this algorithm to kind of overcome those limitations of, say, something like backpropagation, which relies on a convex linear regime. Implementing CMAES allowed me to optimize the network's parameters despite its dynamic structure. Then there was this aspect of spatiotemporal consistency where, we wanted to maintain consistency between the temporal growth of the network and its spatial learning presented. There were some ongoing challenges to this. So the first was that newly added nodes, which were the daughter cells, were positioned correctly relative to existing nodes. We can optimize this process and train the data, we can train the model on the data, but it's unclear as to actually whether we're actually getting the right positions. So this was a challenge, of course. The spatial relationships needed to remain meaningful as the network expanded over time. So we don't want cells that are clearly, say, in the anterior or posterior end of the embryo being flipped or otherwise delocated. And then, of course, we want to maintain those spatial relationships between the anterior and posterior poles, between the left and the right sides, between the dorsal and the ventral aspect of the embryo. And then finally, balancing local and global optimization. Node positions was a constant consideration. So this is where, again, we're visualizing this network, and the network was constantly changing sort of its orientation. So it's kind of hard to see how well it's doing and kind of hard to see the relationship between cells or nodes in this case. And so that's a major challenge as well. So congratulations to Pocky for that work. That was very good, and that was all committed to our DevoLearn repository. And so, yeah, this is a project that relied on 2 open source forks for the TDA or the KMAP or the TDA analysis, and then for this growing networks. We've talked about these source forks, or the source of these forks in the past, so I refer you to those references.
Bradley,
Yeah.
in terms of growing networks, why not make the new nodes a different color and keep the previous diagram and then add the new nodes?
Yeah, I could do that.
Okay.
Yeah.
It seems that might be a way to visualize the growth.
Yeah, that might be an option for. I mean, the only problem there is it gets really crowded, but that's probably okay.
You might have to change this. You might have to change the scale as you go, and then make it into a movie.
Yeah.
It's sort of like the old movies of Hours of 10. Are you familiar with those?
Oh yeah, I remember those.
Yeah, you had to keep changing scale to visualize what was going on. But
yeah. Yeah. Yeah.
I think the problem is we can't visualize a continually changing topology. But if you restricted yourself to the previous topology, when you add the new nodes and make a distinction, you might be able to catch it.
Yeah. Yeah, so it's interesting to note that like the algorithm or the whole approach that was forked from was done with neural networks, and as such, you just basically what they were interested in that is just adding nodes to a neural network, and it wasn't so much important to have like good visualizations of that process because it was just like expanding that network and
Yeah,
kind of
yeah,
using so
yeah. But you're getting the two confusions. One
right, right,
is continually changing topology, and the scale is changing, and so I think in terms of human visualization, we need something to hook onto.
right, right,
And even if it's a changing target, the problem is to smooth out the changes in topology.
yeah, yeah. Taking some notes here. Yep. Okay. Any other questions or comments about that? Okay. So the next thing I'll talk about is Morgan has been posting on some videos, YouTube videos. So we were talking, I think it was last week or the week before, about the Nobel Prize in Physiology and Medicine, and this is, of course, was the prize given for uh, the discovery of uh, I think it's microRNAs and C. elegans. So, they were discovering the function of microRNAs in a very specific context in the up or down regulation of a specific gene, and they were able to do these studies to show this. It was brought to my attention after the meeting that the Nobel was given to C. elegans people, and there's been a lot of work done in plants. We had one paper in the group that I talked about on plants, but they didn't give any award to the plant people who are working on this problem. So it's kind of an oversight, maybe. But anyway, was that?
It's typical.
Yeah. So just so that you know,
Plant
it's
work, yeah, yeah.
yeah,
Plant work in morphogenesis is generally ahead of animal work, but it doesn't get recognized.
yeah, yeah. So that being what it is, we got into this further discussion about like RNAs and the different types of RNAs people have discovered and sort of the functional role of all these RNAs. Some are functional as with very tight targets. Others are expressed, but it's questionable whether they're functional. Others are just present, and so you have a lot of different types of RNAs. You have that they do all sorts of different things in the cell. Sometimes they are involved in gene regulation, sometimes they're not. Sometimes they do other things that are very transient and so forth. So, this is a video from Gary Ruvkun. This is UC Berkeley Events, is the YouTube channel. And I guess he won the Breakthrough Prize in 2016. So, this is his talk uh, for the Breakthrough Prize, The Tiny RNA World, which is just basically talking about the world of different RNA molecules that we know of at this point, and
Yeah,
beginning to
Bradley,
learn about.
there's,
Yeah,
there's an unfortunate there's an unfortunate ambiguity here because RNA world has for the last 50 years or so meant the world when before life started.
right.
Okay.
Yeah,
I know that's not what he means,
right.
but
Yeah, I get it.
he's introduced an ambiguity by using that RNA world uh, expression.
Right. It's the early life clashes with molecular
Well,
biology.
the favorite model for origin of life now is the RNA world, but that means a world in which RNA came first.
Right. Yeah.
Okay, okay,
Yeah. Well, anyways, yeah. So that I mean, he just kind of goes through kind of our state of knowledge there. I don't think he talked. We may talk about the origin of life, but I doubt it. But
Yeah,
anyway, this next video was, of course, Anton Petrov, who's a science communicator. It talks a little bit about the Nobel Prize, how worm mutants led to the 2024 Nobel Prize. So if you go back and read we did it in the meeting 2 weeks ago, where you have two landmark papers in C. elegans where they looked at a specific gene, looked at a specific microRNA, and they were able to kind of get this nail down this circuit where they could see what was going on. So that's like at a more accessible level than like maybe reading the papers actually, because the papers can be pretty obtuse if you're not used to the language they're using, and all of the C. elegans jargon as well. So this is actually, like, Morgan has brought up the Build a Cell seminar a lot. So this is from the Build a Cell seminar channel. This is Anthony Forrester from RNA to Luca Ideas, Data, and Utility. Now, this is actually like kind of more about kind of moving towards RNA world, thinking about like the sort of the origins of RNA, or like we had RNA world, and then we had, of course, complex life, and RNAs, of course, are doing a lot of things. So, what is the connection there? I didn't watch the video, so I couldn't tell you much more about it. I don't know if Morgan could tell us a little bit about it, but it sounds like a really interesting video.
Yeah, sorry. I only saw it last night. And so dropped it in, but like you said, it's always been a great seminar series, so I'm sure it's going to be interesting. Yeah.
Okay, yeah. Well, thank you for dropping those in, and giving us some things to.
I was gonna just say one thing, and sort of, I mean, maybe related to your kind of initial discussion, but just to maybe open it up to any topic. But the Celsius kind of our fearless leader at Celsius was visited Future House this weekend.
Yeah.
And Future House is working on, it's I'm not sure where they got their funding from, but they're funded to create a scientific assistant, and they're doing, so they've got, I mean, I think they've got some various projects, but a couple of their current software outputs are this kind of Q&A assistant that does digestion of kind of particular. If you feed it a collection of PDFs that has some sort of relation or some sort of topic, then it digests those and can produce a kind of Q&A from them, or obviously summaries and things like that.
Right.
And I was wondering if that was if there was already a good kind of curated set of papers on a particular topic that you could think of that might be useful to use as an example. We're going to try and put together. I mean, they're basically asking for an event, and that we can use their space, it's a really cool space down where the kind of new UCSF campus is. So was just thinking about that. Neurotech X. Oh, sorry, daughter was asking me to eat the top of her banana. At Neurotech X, one of the co-founders has a paper on the kind of like a review paper, the literature review paper on deep learning and EEG.
Okay.
And so like he has this great collection of curated papers in a database from, I forget, like 2012 to 2022 or something like that.
Yeah.
And just wanted to put it out there. And, would do we have anything? I was kind of also, not that it's DevoWorm, but thinking about Jesse's paper and kind of review of cybernetics, things like that.
Yeah. Of course, the C. elegans community always has they're very organized in terms of their resources. So I don't know if you go to something like
Yeah.
Wormbase, and they have like
There you go.
they have their sort of source papers because they have all this
Oh
information
yeah,
about different genes, and so that might be somewhere to go. I don't know if they have an API.
Okay. Yeah. This was more like for people to think about, but yeah, that's an awesome one.
Yeah.
And topical.
Yeah. Well, I mean, it's around a certain topic, so like you have like
Yeah.
so you could like discover connections between papers that people wouldn't have thought of because a lot of the genetic work is like on a specific gene, and they're doing a test. They're trying to figure out the function of it and everything. But like people would look across genes and say, in a genomics paper, you wouldn't see this, where you look across genes and you say, oh, these two genes are doing similar things, or usually what you have to do is go in the literature and like pick out basically the genes implicated in function X. It's like if I want to see all the genes that are implicated in like the morphogenesis of the intestine, I have to go into the literature and kind of look for intestine and see all these papers. And I don't know if I have all of the right genes or what. So that would be one really helpful thing, I think.
Yeah, no. I mean, that's interesting. I'm gonna look it up. I'm still working on school run here, but that sounds like it would be a really interesting. So is that a database or?
Yeah, it's Wormbase, and so they have a lot of community resources. So
Got
yeah,
it.
Wormbase is here. You have basically this version of the genome, C. elegans genome, and you have the nomenclature for the genes, and then you might search for a gene, and it tells you kind of what it does. So let me see, I can find one here. This is AAK1, and it has like the different information about it and tells you where it is in the genome. And I think there are papers that are associated with this. I mean, I'm not sure. Yeah, I'm not sure if they have them because they have to get their data from somewhere. So I'm not really sure how you would access that, but there are usually papers associated with each of these genes, like where they actually found them, or studies that talk about them. So it may not be Wormbase that would be the best source for the data, but it would be like maybe PubMed, and then you could validate it with Wormbase.
Well, yeah. I mean, I see this genetics paper 2022. Wormbase in 2022 data processes and tools. Yeah. Well, it's still really interesting. It would be obviously a very, very good topic given this meeting.
Yeah.
But again, perhaps a good topic in terms of what Future House's goals are.
Right.
Because again, I think that is their goal, is to create the kind of topical a topical article.
Yeah.
Didn't mean that to rhyme, but yeah, that would be interesting.
Yeah. So why don't we? Yeah, I think Jesse put the link to Future House. So why don't we take a look at that? If
Yeah. And so you can check out the tools section. I don't know if they've got an about, because honestly, I don't know their history.
Oh, the about is we're building an AI scientist. And
Yeah, I was hoping for a bit more from the about.
So yeah, well,
Because I mean, somebody gave them a lot of money to set up.
yeah,
And the other thing that came up in our meeting was that they had bought their building, because there's a bunch of buildings in San Francisco that are going really cheap, again, relative to their last sale,
yeah,
right? Because especially commercial real estate is kind of cratered here.
yeah.
And but that's still a lot of money.
If you have a human, it's the quest. Then AI scientist, you have a world model, hypothesis generation, then experimentation. You have this AI science assistant, agents for specific biological workflows. So you could have like for next gen sequencing or some literature search. And then they have AI tools, which is the predictive type model, which is like AlphaFold, APIs, which allow you to get your data set, and then laboratory experiments, which are, of course, you know, drawing from the empirical stuff. So this is kind of like, like the community tool I showed you, Wormbase, has like the API and the laboratory experiments. They just need the predictive model aspect plugged onto that to find relationships between genes or something that, because we do a lot of genomics, next gen genomics, we get a bunch of data, but, the whole point of that is to synthesize it and find insights. But oftentimes we just, see like some sort of UMAP and then people make statements about it and that's it.
Yeah, sure.
Well, Bradley, I've got two questions.
Yeah.
Has the AI scientists published any papers yet?
No.
What was that?
Well, like again, there's so far they've only got these two tools. It's like the QA summary tool, and I think that's just their hypothesized design.
Well, okay.
Yeah.
So this is very far from an independent scientist.
Yeah. It's like I've
Okay.
read about this where a lot of people are trying to build like what they call an AI scientist, basically using an AI to do like this type of synthesis that a scientist would do after they've run a bunch of experiments. How would they find the insight in what they're doing? And
Yeah, but has it submitted any papers and passed peer review?
no, no, it's all vision. And in fact, there was a paper that I had in one of my classes that we went over that was like basically the argument, they made the argument against the viability of an AI scientist because they don't have the sort of like when people are trained as a scientist, it takes a certain amount of time. And it's not that you're acquiring skills, but you're also acquiring this intuition for science. And so that's a thing that an AI scientist is missing specifically. So they were arguing that that's not going to be an easy thing to overcome. But yeah, it's a pretty visionary.
And Blazek at Argonne has like much more practical work, systems that they've been developing for. That they've been developing for material science.
Yeah.
So this is, and it'll be interesting to see their output of their last kind of meeting / hackathon.
Yeah.
So I mean, again, it's like one of the reasons, it's not just the agent, it's the agent's access to information that'll make it, in any way practical.
Right.
And what Ben's trying to do is definitely like, it's like, yeah, kind of like automating design of experiments. I think it would be a good way to put it.
Yeah.
Like, it's not so much a scientist, it's just like, hey, we've got to cover this multidimensional space.
Right.
And yeah, it's like kind of some basic planning, as opposed to any kind of like, AI scientist.
Right.
Yeah.
I mean, like on Saturday, we talked about the AI of
You
the
see,
1970s and how like they thought they had it in the bag with respect to like planning algorithms and other things, and then there was this huge like winter where all of that kind of went away, and they started on the path towards statistical AI, or using statistics more than like rules. So it was very interesting because it was like they were talking about some books that were written right at that time, and people were like really excited about things like having an AI scientist, where you could, use rules to do things that say a scientist would do, but that never came to fruition. So
what salary does an AI scientist demand?
initially nothing, but who knows?
Okay, they're
Unlimited compute resources.
Yeah,
yeah. Except for that, you see, all support for scientists can go away, and so all the grant agencies can disappear,
yeah,
putting us all in poverty. The other thing is, it seems to be confined already to a certain database, and therefore, getting AI to make a transition to a different level of thinking would seem difficult.
yeah,
Now, the trivial one that comes up is the one we work on, which is the transition from genes to differentiation trees. Could AI ever make that transition?
I don't know. Yeah, I mean, like it. I mean, basically, it it does rely on the training of the model. So they talk about a world model too, which is of course this idea that you can build this model of the world that gives you enough information to sort of represent any, like if I train a model, it can take that data and spit it back out to me. But like, does it have this sort of conceptual framework that if I give it any data set in biology, well, let's say genomics or genetics, but any organism, like it could be bacteria or plants or animals, could it give me a reasonable answer given that it has a general sort of framework for life, what to expect, and what the rules of life are, which humans don't even know. But let's suppose that we could sort of get it to learn this world model of biology, or at least genomic biology or genetics. So that's what you would need. It wouldn't just rely on the training data sets. It would rely on this world model, and that's going to be probably the thing that does not make it happen. But who knows?
Yeah, maybe they should call it AI librarian, because
Yeah,
it basically, in the good old days before AI, if you had a problem, you could bring it to your librarian, who would do the lookup service for you.
yeah,
Okay, so it seems to me they're replacing librarians more than they're replacing scientists.
yeah. Well,
Well, the tools that they have are very modest.
yeah,
I mean, if you look at them, and AI librarian might be actually the best, AI research assistant or something like that, which is kind of the librarian function you're talking about.
Yes.
yeah.
uh, um, is kind of what their QA tool is, you know, not even like reporting to do. It's just, you know, it's what they're trying to do. Yeah.
Yeah, I have it up here. So there's Paper QA, which is one tool. This is a minimal package for doing question and answering from PDFs or text files. So this is where you train it on PDFs and you get like this answer. It strives to give very good answers, no hallucinations, by grounding responses with in-text citations.
In other words, preserving the hallucinations that scientists have.
Well, yeah, it could be that. And then, yeah, it could be an internet rando, you know. But yeah, and then Hassani one, which is hassani1.com, a minimalist AI tool to search if anyone has ever researched a given topic, which is, you know, I mean, that's within the bounds of the published literature, but trying to find like holes in the literature, basically. So, um, yeah, I mean, you know, this is an area where, again, it's going to depend on what it's trained on, what, you know, and then, but, you know, the tool itself is actually a nice sort of summary. And I think that's where the power of this is sort of in this librarianship or being able to help
Yeah.
research the literature.
Yeah, I'm not arguing against it. I'm just saying that it's rather presumptuous that it's a scientist.
Yeah. So yeah, Morgan, is this like what is this based? Is this are these large language models or are these like other types, different types of models that?
Oh yeah, no. These are large language models.
Okay, yeah,
The only thing I was gonna maybe say is that, you know, when they say world model, you know, I think they're more, you know, I'm just thinking about those papers that Saffron had in his RFP. Those kind of early, like, I want to say like 2014, like 2016 kind of papers. I'm trying to remember the group, but it's really just about like trying to have some sense of planning.
yeah,
Yeah, like and yeah. So my daughter is telling me what I need to pack in her bag. Just so, but yeah, yeah. Anyway, it's very rudimentary. Or, you know, again, it's somebody trying to take large language models and connect all the different kind of, you know, specialized features or kind of functions that you know somebody has done research on, and then put them all into one graph, right?
yeah,
And then say like, we need something like this. and certainly, you know, like it's, you know, archive sanity preserver came along because, you know, machine learning researchers were being completely overwhelmed by the amount of literature that was coming out, right?
yeah,
And like, you could almost say the same right now, just about bio or med related LLMs,
yeah,
right? Just that specialized topic, you know, and um, yeah, yeah. So it's yeah, yeah. So it's definitely like it, or thinking about. I mean, I also think about that that project that just like knew the people at like the MIT Latino computer science, you know, that made, you know, the connections between literature.
okay,
I can't remember now.
Oh, the state of the art stuff. Yeah.
Yeah. You know, it was there are all these tools that have come and kind of come and gone, unfortunately.
Yeah.
But that were really helpful in terms of I mean I routinely now use the PubMed's similar and connect cite certainly the citations, right, and that's the old Web of Science approach, but similarity measures can be really interesting, and other other literature helpers. I don't know what to call them. Sorry, I'm trying to multitask here.
Yeah. Yeah, I think that's yeah, there are all sorts of tools that kind of come out and
It's the
algorithm and Dick, you were gonna say something?
oh, I'm talking to my dog. Sorry.
Oh, it's all good stuff. Yeah, I'm glad we brought that up because it's I think really interesting stuff. Kind of how these different tools are evolving, and in C. elegans, of course, we have this very rich community of kind of data and resources like that, and sharing of things, and so this is actually maybe a next step in that. And I don't know if OpenWorm has really experimented with this too much, but it might be kind of an interesting kind of OpenWorm project, because I know we've done like things in OpenWorm with the literature and some other tools, but nothing like an LLM or anything like that, where you're actually trying to train a model to give you you know generating something that can be useful for research. So
Yeah, if it's useful, maybe Elon Musk will incorporate it in his robots.
yeah.
It's called a lab assistant.
Yeah, well, yeah. Okay. Well, do we have anything else we want to talk about before wrapping it up for today? Or I think we had pretty good discussion here. So yeah, yeah. Well, thanks for attending, and see you next week.
Okay. Take care.
Take care.
Bye.
Thanks. Bye.
Right.
Bye. Okay, so our final discussion during the meeting was about using world models and other types of training data to build this AI scientist. And what's interesting here is that, OpenWorm, we have a lot of data sets. More generally in the C. elegans community, we have a lot of data sets that are shared, that are available to, do things like data mining or to build models from. But one thing we haven't really addressed is this idea of a world model. So I'm going to go over quickly about maybe the outlines of an OpenWorm world model, or maybe a world model for C. elegans. Maybe, if we talked about sort of these general biological models or biological world models that you might need to do this sort of AI science across species, but focusing just on C. elegans, I think we can see the pieces that need to be in place, at least to get to a starting point on this. So the proposed OpenWorm world model would consist of at least three major components. The first is mining, or at least capturing all of the C. elegans databases and using those as training data for this world model. So as we showed in the meeting, we have genomic data, we have different experimental data, we have morphological data, we have data on the connectome, we have electrophysiological data. These are all things that are available to us through, the community. OpenWorm has curated a number of these data sets, as well as, some of the other things that exist in the biological community at large. So if we go to GenBank or NCBI, we can find a lot of these resources as well. Basically, training on as much C. elegans related data as possible. Then we need general models of sort of biological processes. So these might include models of morphogenesis, genetics, metabolism, connectomics, electrophysiology. We might also have a general model of development since we have an entire lineage tree worked out for C. elegans. We might have a general model of behavior as well. And so we have a lot of those types of data. Like we have worm movement data that is sort of, unclassified. We have a lot of raw data, video data. Those all could be built into this general model. But, setting kind of behavior aside, we can then build functional models. So in this case, I say here that functional models are things like movement behaviors or social behaviors. Functional models really would be anything that tells us something about the specific function of cells, of organisms for different sort of domains. So we have the general model of, say, morphogenesis or metabolism or genetics, which would just basically be, what kinds of circuits do we have, movement circuits, what kinds of gene regulatory networks do we have, what kinds of other types of networks do we have. We basically want to describe these processes in C. elegans at a very general level. So, for example, with morphogenesis, we have specific things in C. elegans like movements in the embryo or patterns of division in the embryo or patterns of differentiation in the embryo. We can see this in the lineage tree. We have patterns of connectomics. So the C. elegans connectome is unique from other connectomes. It's also unique from a random graph. Those general models give us these tendencies or this sort of structure on which to hang any sort of prediction from data. And then the functional models would be more about sort of a dynamical model or a time-dependent process. And so, it could be like social behaviors, like, there are different social behaviors C. elegans engages in, or movement behaviors that C. elegans engages in. We know that the movement, C. elegans movement repertoire is a somewhat low dimensional space compared to something like the genomic space for C. elegans. So we can characterize all these functions aside from sort of these general tendencies, and having models of both of those would contribute to a world model that would then allow us to, basically train the model, have this world model, and then be able to query the model for predictions or insights. So, that's something that I just kind of sketched out from our discussion. Hopefully, we can kind of clarify this a little bit more. We might actually put together a preprint on this. So stay tuned.